flower to pollen reaching toward the other flower stigma, that is cross pollination. Self pollination, cross pollination. Types of the self pollination, types of the cross pollination, reason for the cross pollination, that all we will discuss. Very, very important. Each one is very essay question. Very, very important. Very important point of view. These all are very useful to identify, to know, and to learn the things. These chapters are very, very useful to learn the life cycle regeneration of the new plant from one plant to other. Seed formation. Okay. And the fertilization process. You know the fertilization in angiosperm, double fertilization will take place. Whereas the gymnosperm only only will take place. Only syngamy will take place in the gymnosperm. You see, female gamete and to subsidiary cells. You know the 8 nucleus 7-cell gamete. 8 nucleus 7 cell. You see, above 3 are operators, below 3 are called as powers, and the middle 2 is called as polar nucleus, or also called as secondary. Okay. These X cell to subsidiary cells, 3 cells, the X cell operators. If the pollen reach toward the stigma, the pollen reach toward the stigma, and then it goes inside through the style and reach to the ovule. In that, it reached toward the female gamete, toward this side. The germination of the pollen tube. The pollen tube germinates and it reaches toward the egg cell. And first male gamete fuse with the egg cell. That is called as fertilization. Syngamy, fusion of male and female gamete, is called as syngamy. That is also called as fertilization. Have you understood the difference between the pollination and fertilization? Pollination means reaching the pollen toward the stigma. Up to here only it's called as pollination. After that, fusion of the male gamete with the female gamete. Sperm cell with the microspore with the egg cell. That is fertilization. Here, the dicot, what happening? The two male gamete. First one is mating with the egg cell and it forming the zygote, embryo, diploid embryo. And the second male gamete is fusing with the polar nucleus or secondary nucleus. It having the two nucleus, diploid. It is diploid already. Diploid plus one male gamete. So that it will become triploid. The triploid nucleus will form. That we call it as pen. What we calling it as? Pen. Pen means not writing pen. Okay. Pen means primary endosperm nucleus. Primary endosperm nucleus. That is forming by the secondary petalization, not primary pet. By the primary petalization, embryo is forming, diploid. By the secondary fission, that also we also call it as triple fission. Why we are calling triple fission? 2N, 2 nucleated polar nucleus plus 1 nucleus, second male gamete. That means it is forming the 3 nuclear structure. That's why 3 nucleus are fusing. That's why we call it as triple fission. Triple fission. We are calling it as triple fission. This first identified by Navasin. Who is that? Navasin. And first petalization syngamy first identified by Strasburger. Pollen germination and pollen tube formation reaching toward the egg cell, reaching toward the female gamete, and there the microphyllum is reaching toward the egg cell, fusing the male and female. That is called as syngamy. That first identified by Strasburger. Strasburger, we call him as father of embryology. Who is the father of embryology? Who is that? Father of embryology. Who is the father of embryology? Strasburger. First identified the petalization process. First discovered. Okay. And you know who is the father of Indian embryologist? Who is the father of Indian embryology? Embryology from this form? P. Maheshwari. Panchanan P. Maheshwari. Maheshwari means not lady, not girl, not woman, men. He is the scientist, embryologist. He has done so many embryological works. He made artificial fertilization. He known the fertilization process techniques, parthenocarpy techniques. Has written the book also. P. Maheshwari, Panchanan Maheshwari is the father of embryology of our India. Indian father of embryology is Maheshwari. Overall father of embryology is Strasburger. This you should remember. Okay. And then pollination, fertilization. Whereas in the gymnosperm, I told you that in angiosperm, 2 times fertilization. 1 male gamete fusing with the egg cell, second male gamete with the diploid nucleus. Triple fission. Syngamy plus triple fission. Syngamy plus triple fission. Commonly called as double fertilization. Double fertilization. In angiosperm, double fertilization is taking place, whereas in the gymnosperm, only 1 male gamete, only 1 male gamete that is fusing with the egg cell. Before the fusion of the egg cell, male gamete with the egg cell, whatever it is the female gametophytic tissues that taking mitotic division, not the meiotic division, not the reduction division, somatic division. Mitotic division will take place so that single nuclear haploid nuclear cells, multi number of the haploid nuclear cells will be formed. Multi number of the haploid nucleus will be formed that are in haploid. That's why gymnosperms endosperm is haploid, whereas the angiosperm endosperm is triploid. That you should remember. In any competitive exam, they may ask question. Why the angiosperm endosperm is triploid? Endosperm is forming by the triple fusion of the three nucleus, secondary male gamete and polar nucleus, secondary nucleus. 2 plus 1, 3. That's why triple fusion. By the triple fusion, 3N endosperm is forming. That all the techniques also we will discuss. And the endosperm types, endosperm types also we will discuss. Cellular, nuclear, helobial, ruminate, endosperms that we will discuss. Embryos types also we will discuss. Dicot embryo, monocot embryo, and embryogenesis and self-incompatibility. Why in some plants from the same plant pollen is not germinating? That is because of that is a hidden reason, self-incompatibility. If the pollen came from the same plant, it will not germinating, it is die and drying and diluting. That because of hormonal reasons, it is rejecting their own pollen, accepting other flower pollen of the same genus, same species. That self-incompatibility and parasexuality also we will discuss. Parthenocarpy, that also called as parthenocarpy. Without the fertilization, without the fusion of male and female also forming the fruit, that we call as parthenocarpy. That all we will discuss. Okay, these all are the overall, apomixes, without mixing of the male and female. Apomixes, without mixing of the male and female gamete, the embryo formation is taking place. Apogamy, apospory, that we will discuss. Apomixes, important question. Parthenocarpy also. Without the fusion, without the fertilization, fruits are forming, either from the sexual organs or from the even from the somatic organs, somatic parts, vegetative parts. That is happening. That also we will discuss. Parthenocarpy. Okay, and importance, embryology in relation to taxonomy, in horticulture, in agriculture, how we are using embryological techniques, embryology knowledge, embryological science in agriculture, horticulture, in the taxonomy, how embryological character are basics importance. That also we will discuss. That also very, very important. In the taxonomy, embryology is the basic part, the foremost first distinguishing characteristics to identify the plant, to distinguish the plant, to variate the plant. The embryological character is very, very important. Even 3 types of the classification, classification type, artificial classification, natural classification, phylogenetic classification. The 3 classification, we follow the floral characters. We depend upon the floral character. Even artificial classification also, Linnaeus. Who is the Linnaeus? Carolus von Linnaeus is the father of taxonomy. Father of plant taxonomy, the Carolus von Linnaeus. Linnaeus binomial nomenclature. But the binomial nomenclature first proposed by Gaspar Bahin. But later it is strictly implemented after Rivinus rules. Only the two epithets must be there, not more than that. If the more than two epithets for each plant name, it is difficult to remember, difficult to read out, difficult to know the sharing knowledge. That's why Linnaeus given followed the strict binomial nomenclature, given the names to so many plants. Otherwise, earlier the plant name is very lengthy. Veera Venkata Suryavara Prasada Rao, like that. You see, Polyalthea Coromandeliana, Polyalthea Coromandeliana, Acuta, like that. The much more lengthy name. Albus, all the characters showing showing in the name. That is very hard, difficult to remember. That's why, as for ICBN nomenclature, only two epithets must be there. That we will discuss later in the last chapter. Okay, now the first chapter is anther. Anther are also called as microsporangium. You know, anther, that means androsium and gynosium. Gynosium and androsium. Gynosium and androsium. These two are essential organs of the flower. And sepals and petals are the non-essential organs of the flower. Sepals and petals will be useful to attract the flower, insects, other birds, animals. Only for the pollination it will be useful. After pollination, there is no use of the sepals and petals. Sepals and petals, the petals will be fell down, or dried, or dropped. But the essential organs, gynosium and androsium, is very, very important. Androsium, anther, up to releasing of the microspore, that means pollens, that will be useful. Whatever the pollens released, that reach toward the stigma of gynosium, and through this style, it will go toward the female gamete. That type of the fertilization, we call it as siphonogamy. Siphonogamy. That means male gamete is going toward the siphon style, tube-like structure, toward the egg cell for the fertilization. That's why we call it as siphonogamy type, siphonogamous type of the fertilization. Okay, siphon means tube. Here, male gametes are moving from the tube, from the style, from the stigma to egg cell, egg cell. That is called as fertilization. Okay. Here you can see of the two essential organs, one of the essential organ, we calling it as anther. We are calling it as anther. You see, we totally we calling it as androsium. And the androsium, two parts are there. First one is filament, second one is anther. Sac-like structure is calling it as anther, and stalk-like structure we call it as filament. These two commonly called as androsium. These two commonly called as androsium. Here we are going to discuss this anther, male gametangium, also called as microsporangium structure, and microsporogenesis. That means pollen formation of the pollen, and special type of the microsporangiums, especially in the cypress type of the microsporangium, anther, and a Nemak phenomena, the second one. Anther, that means microsporangium will look like a megasporangium with the 8 nucleus, and it will be acting as a female gametangium. That we will discuss. Okay. Now, in the androsium, anther, and filament. Here the bilobed anther. We are having the 2 lobes, but it partially lobe so that it's forming the 4 lobes. 1, 2, 3, 4. 4 lobes are present. If we take the section of this anther, it will be look like TS of the anther. Yes, a question. TS of the anther. Very, very important. Yes, a question part of 2. And the tapetum. What does it mean? The tapetum and tapetum types. 2 types, amoeboid and glandular. Tapetum. 2 types of the tapetum. The tapetum and tapetum type and tapetum function. Tapetum function as a nutritious layer to supply the nutrition to the maturing pollens, mature microspores. To supply the nutrient to the mature microspores is the main function to get mature the microspores. Okay, that is the main function of tapetum. It is a nutritious layer, innermost layer. Okay, we will discuss here in this continuously. We are we are following in this. Okay, you see how the anther structure is. It is tetralobed structure. How many? 1, 2, 3, 4. 4 lobe structure is there. And each lobe, if you see each lobe structure from upper side, from top to bottom, there is a parts. What are those? What are those? Uppermost layer, we call it as uppermost layer. We calling it as uppermost layer. We calling it as epidermis. Okay, is a single layer without any gap, tightly coiled cells. It covers all the androsiums and it gives the specific shape and structure to the anther. That means microsporangia. Microsporangia means it having the microspores, small spores inside the sac. That's why calling it as microsporangia. Okay, that having the layers. Outermost layer is epidermis, single layer. The next one is endothecium. Second layer is calling it as endothecium. You can see here the uppermost layer is calling it as epidermis. The second layer, uppermost layer is the epidermis. The second layer just below the epidermis. Maybe one to two layers, 2 to 3 layers is present. That we calling it as endothecium. Second layer is called as endothecium. And the 3 to 4, 5 layers that are we calling it as middle layers. We calling it as middle layers. And the innermost layer of the the anther is calling it as tapetum. Calling it as tapetum. Okay. The middle layers class. Middle layers, 3, 4, 5 layers. Middle layers are there. That middle layer cells in the third part. And then the innermost layer of the anther is calling it as tapetum. Just below the tapetum, the microsporangium spore sac is present in the microspore mother cells. Microspore mother cells, MMC. Microspore mother cells are present. Microspore mother cells, these are diploid. Microspore mother cells, these all are diploid. The diploid cells, it take the reduction division so that it form the haploid spores, that we call it as microspores. Also we call it as pollen grains. More number of the pollen grains will be formed. For that pollen grain formation, the tapetal layer will be useful. Tapetal layer, it supply the nutrition. Nutrition supply will be done by this tapetal layer, nutritious layer. Tapetal layer also called as nutritious layer. It is the innermost layer of the anther. Okay, that is very, very important. Okay, and here you see in between the two lobes, there is a connecting tissues which connect the lobes of the microspore mother cell, microsporangiums. Okay, this is the basic structure, and each and every part we will discuss here. You can see how it is. You see layer of the microsporangium, it forming the microspores, sporangial tissue, microspore mother cell, that releasing the microspores. You see how it is. You see microsporangium, in that it having the microspores. Okay, you see the stomiums where there is a stomium. Okay, stomium where there is a connecting link here. That part we calling it as a stomium. You can see the stomium part. You know, the stomium part is a very, very important. From the stomium cells, where there is a thin coated cells, that thin coated endothecium cells we call it as stomium. From there only the spores will come out. Okay, and the next is middle layers. You see the middle layers. And the innermost layer is the tapetum, nutritious layer, and inside microspore mother cell. Inside microspore mother cells are present. You see the each sporangium how it is, how it look like. The middle layer, endothecium, epidermis, strobium, microspore mother cells. These all are there. You can see clearly. Conjunctive, connective tissue, which connect the pollen sacs. Okay, and then you can see the angiosperm flower. I already showed you the anther, microsporangia. Sometime, usually, each anther, microsporangia, I already told you, having the two chambers, bilobe. But in the Malvaceae, only one single chamber anther is present. In the Malvaceae, single chamber anther is present. You see, in the Malvaceae, only single chamber, unilocular anther. Whereas in the most of the other plant, the anther is bilobe. Bilobe. Each lobe each lobe it having the again two lobes, so that four lobes. If it is a single lobe, it will be it having the two lobes. The single lobe structure is there in the Malvaceae, whereas the remaining or majority of the plant having the bilobe. But in the Bixaceae, 8 lobes are there in the Bixaceae. Okay, 8 lobes are present. Bixaceae, Lorantheaceae, and even in the Pitcolobium. You see how the structure is from the outer to inner layer. Outermost layer, I already discussed now. Outermost layer is calling it as outermost layer is calling it as outermost layer we calling it as epidermis. Outermost layer is calling it as epidermis. And just below the epidermis, you see 1, 2, 3, 4 multiple rows of the tissues which give support, strengthen, and uh, prevent from the damage of the microspore mother tissues. Okay, and the next is middle layers in between the endothecium and tapetum. The endothecium, that is a tapetum. In between the endothecium and tapetum, there is a middle layer, 3 to 4 layers of the middle layers, and then the tapetum is innermost layer. Innermost layer of the anther is called as tapetum. And also it is a nutritious layer. It supplies the nutrition to the maturing spores where it is getting mature. That all the maturing spores will be given suggestion, guide, so that it will be almost all the spores will be mature and germinate and take the fertilization. Okay, that is very important. And then here you can see how it is in the cross section. Actually, how the microsporangium is taking place, developing archisporium because having the much reproduction with the means anther lobes. Already told you which is right. Many of Loranthus is only one, and majority is the two lobe, and in Malvaceae only one lobe. Here you see the development of anther, how it is taking place. Anther, that's the main primary anther, primary archisporial cells. Primary archisporial cells. Primary archisporial cells are modifying primary archisporial cells that are being modified, and it is forming various tissues to protect, and some of the tissue as a germinating tissues. Here you can see the archisporium or primary archisporial cell. Archisporium or primary archisporial cell or archisporial mother cell. Okay, that is dividing into two. First one is epidermis, second one is archisporium. You see epidermis and archisporium. Epidermis is the outermost layer, extra outer layer. It gives envelope and it gives strength, support, size, shape. And the second cell is being modified as an archisporium. That means as a generative cell. The archisporium, primary archisporial cell dividing into secondary archisporial cell, and archisporial mother cells will be formed. From that, microspores fertilization. This microspores will be formed. Okay, and in the archisporium, I already told you again two types of the cells are formed. That is primary archisporial cell and primary parietal cell. Parietal means peripheral, partial, outside. Primary parietal cell and primary archisporial cell. Okay, primary parietal cells again divided into secondary parietal cells and secondary two secondary parietal cells. That you see the outer parietal secondary parietal cell is forming the endothecium. Endothecium, and you can see some of the tissues of the primary endothecium. They are giving the space to form the multiple layers. To form the multiple layers. That is the reason. You should otherwise you may confuse. That we have to follow the modern trend or old trend. What way we have to identify the plant. Only strict thing is you should know about the plants, their life cycle. Okay, and then you can see here the first one, primary archisporial cell divided into two. That is primary parietal cell, primary archisporial cell. Okay, and again primary archisporial cell divided into two. That is secondary parietal cell 1 and 2. The secondary parietal cell again it takes the division of into 2. Okay, and you can see this outer layer is forming the endothelium, and the remaining 3 cells are forming the middle layers. Forming the middle layers. Even upper secondary incidents also forming, so that it is forming the epidermis, endothelium, middle lamellar, unilamellar, DNA, whatever it is. Okay, and then the next is mitochondria. Okay, you can see here how it is. The innermost layer of the secondary parietal cell is forming. What it is forming? Tapetum, the most important. Tapetum, the most important tissue, and the innermost layer of the anther. Innermost layer of the anther will be formed. Okay. And next, what are there in the secondary cell? That we call it as primary sporogenous cell. Primary sporogenous cell will become the secondary sporogenous cell. Secondary sporogenous cell, secondary sporogenous cell will become the microspore mother cell. Will become the microspore. It will become the microspore mother cell. Okay, primary parietal cell. Primary parietal cell divided into 2 and again 2 into divided. And secondary sporogenous cell are sensitive so that this microspore mother cell will take the reduction division and forming the applied spores called for the employment, the new employment. Okay. For the fusion, for the pollination fertilization, it will be useful. Okay. And then here you can see going on from up to the maturation. What are the tissues, modifications, developments are occurring? The microsporogenesis. That also question will come. Microsporogenesis, formation of the microspores and from the anther. That means anthria. That means male gametes. Formation of the male gamete. Generation of the male gamete is taking place. Here you can see the endothelial cells just below the epidermis. Endothelial cells up. Fibrous bond. It looks like a fibrous brand. It gives the support, strengthen, elasticity to the whole anther. This is all we discussed. Epidermis is the outermost layer, protective it will protect. And here in the epidermis, very thick coated cells are there here and there. The thick coated cells, we call it as stromium. I already told you the stromium cells. From the stromium cells, the spores will come out. Okay. Middle layer and microspore mother cell tapetum. These all are the parts. You see what is the function of endothecium? You know what you should remember here is the most important. Innermost layer and nutritious layer supply the nutrition to the whole plant. And not all plant. If see, you can see here this nutritious layer. This nutritious layer, it is having the fibrous, thick-coated structure organism that you can see here. The microsporogenous tissues are inside, which is covering by the tapetal layer. The tapetal layer immediate to the microsporangium. It gives strength and support, safe structure, and also supply the nutrition toward the developing, maturing microspore, that means pollen grains. Here you can see the two types of the tapetum. That is very, very important. What is the tapetum? What is the functions of the tapetum? And types of the tapetum. You see how two differences. What are those two differences? Okay. Glandular tapetum and amoeboid tapetum. You see in the glandular tapetum, endothecium, middle layers, all are in the unique row, specific row. Rows, you can see the rows, there is no damage. Whereas in the amoeboid tapetum, Plasmodium tapetum and amoeboid tapetum, what happened here? There is no tissue covering cells, envelopes. All are get damaged. All the cells will be come at the middle. All of the cells will be come at the middle, and it supplies nutrition. Okay, these are the two type of the tapetum. That is secretory or parietal or glandular tapetum. Secretory, parietal or glandular tapetum. Secretory, parietal, or glandular. That I told you. It secretes the nutrition and it is not splitting any cell wall cells. It's within the inside. Whereas the amoeboid are invasive or periplasmodial tapetum, it splits all the tissues and all the cytoplasm will come at the mid will come to the towards all do the protection and it supplies the nutrition to mature the young pollens. Okay, these are the two types of the tapetum. Types of the tapetum is very important, and TS of the anther also very important. These are all the parts you have to write. You see amoeboid tapetum how it is, how the amoeboid tapetum or periplasmoid tapetum. Here you can see epidermis endothelium. You see the tapetum especially. The tapetum we calling it as glandular septa, heterogeneous cavity. Okay, glandular septas are present. That we have to see and survey. And the second one is secretary or secretary. Okay. Next one is secretary type. It splits their tissues. It splits their tissues, cell walls, and so that all the cytoplasm will be enter into the microsporangium, so that the cells will be cells of rupture cells. Nutrition will be utilized by the microspores which are maturing. Okay, these are the two type of the Ubisco bodies are present in the amoeboid tapetum. Okay, Ubisco bodies are present which are support give the extra supporting. The Ubisco bodies are helpful in the formation of pollen wall. The pollen is the one of the most prestigious and most strong living plant pot. It will be remain for long time. What's the reason behind it? Do you have an idea? Because of it having the sporopollenin at the outermost layer of the pollen. If you take the pollen, two layers, outer, exine, and intine. The exine is made up with sporopollenin. The sporopollenin secretion is done by Ubisco bodies. This sporopollenin will not be dry, will not be stiff. It will be very hard coated layer, so that the pollen will be remain even in the hot, dry condition, even in the water, even in the air atmosphere. It will be remain because of this hard coated spore. That is preparing by these Ubisco bodies. What that means, where there is a tapetal cell sap, there it is forming the Ubisco bodies. That Ubisco bodies are useful in the preparation of tough layer of the pollen. Okay, that's very important. Sporopollen. That's why the pollens are hard, tough. They don't get damaged. Okay, these are the most functions I already told you. Functions of the tapetum. It will be useful for the development of pollen and also for the transportation and also for the maturity and also Ubisco bodies I told you to form for the synthesis formation of the sporopollen and tapetum. Tapetum will be degenerate if it is a pollen will sterile only. Otherwise, it will give the supply the nutrition. And the callose plugs are there in the pollen wall that gave the cytoplasmic male sterility. You know the gametophytic male sterility and cytoplasmic male sterility. Two types are there. If it is sterility is causing by the stigma ejection, that is gametophytic, or otherwise if there is no fusion is taking place, that is by sporophytic cytoplasm revolution. Okay, you see in the microsporogenesis, while forming the pollen, what is happening here? The formation of applied microspores. Usually, it form the from the where the microspore mother cell will take the reduction division and it form the 4 applied microspore. Okay, that you see there is a 2 types, simultaneous type and successive type. These are the 2 types of the pollen bunch formation is taking place. First one is simultaneous. That means if the one cell is dividing into 2, immediately cell wall also will be formed. Successive. Yes. And then the next after second division, it forms the 4 cell wall so that it will become the 4. Another one is the successive. After taking the 4 division, forming the 4 cells only, it forms the cell wall. These are the 2 types, so that it forms the various structures, various type of the structure of the pollen. You see the various structures of the pollen. You see the pollen tetrads, 4 microspores from the pollen mother cell arranged in various structures. You can see tetrahedral, 4 spores in 4 sides. కనిపించని 4 సింహం 4 spore, 4th pollen, the back side. Okay, and isobilateral pollens, two pollens are side by side, and decussate, 2 pollens are in horizontal, 2 pollens are in vertical. 2 pollens are in horizontal and 2 pollens are in vertical. You can see of the pollen, 3 side and 1 maybe back side. Tetrahedral. You see isobilateral, 2 one side and 2 other side by side. And in the decussate, 2 are in horizontal, 2 are in vertical. 2 are in vertical, 2 are in horizontal. And T-shape here also, but micropylar end one is in horizontal, chalazal end one is in vertical. And linear one is 4 microspores arranged in one line. 4 microspores are arranged in one line. And you know, these all the 4 types also will be available in the power plant. That is very, very important. All the 4 types are present in the single plant. And then along with that, not all the pollens are arranging in the pollen tetrad. Other types of the polysporia also is there. You see Hapnia indica, compound pollens. In mimosis, albizia, mimosa. You see acacia pollens. You might have seen the acacia pollens. You see how yite structure. And you might have seen. Hello.
Hello, sir. Time up, sir. Next class need to start.
Time up. Just ఒక 2 minutes లో complete అయిపోతుంది.
Okay, sir. Okay.
Just two అయిపోతుంది.
Okay, sir.
And just you can see here, there is another type of that one you can see in the Asclepidaceae family, Pollinias, of the pollen sacs. Is combined type. If the pollen sac, the two type of the pollen sac, each having the pollens inside, it having the more number of the pollens. There is advanced type of the pollens formation, which are there in the two families, that is in Asclepidaceae and Asteraceae. And one more is there, another very important one. You see how the microspore mother cell is taking the divisions. You see in the majority of the angiosperms, usually in the two male gametes will be formed in the totally in the three nuclear stage, the pollen will be come out. Of that, the two are male gametes, one will be the two nucleus. The tube nucleus that will be useful for the formation of pollen tube, germination tube. Okay, and then are there whenever the pollen is you can see generative cells. You see the vegetative cell again dividing and it forming the vacuole and tube cells, and the generative cell again divide into whatever the two that are will become the two male gametes. This cell another one with the polar nucleus. That's why angiosperm, endosperm is triploid. I already told you. This is also one of the four most important, very important in the competitive exam point of view, your knowledge point of view. Cypress type of the pollen. Usually pollen, how it is, it round, circular, it having the four pollens inside. But here you can see the four of the four, three is in one side, only one will remain, one will be the larger, and the remaining three will be very small, minute. This all will going to degenerate. Only one pollen, one nucleus will be remain. This is there in the Cyperaceae family. There is the advanced type of the pollens. Only one pollen within the forming from the pollen mother cell. The very important. And then another most important is Nemak phenomena. This is also very important in competitive exam in short answer. These all very important. Cypress pollen, Nemak phenomena. These two in the short answer point of view also very important. Even in the competitive examination point of view also very important. You see first identified by the Nemak, that is in Hyacinthus orientalis. Hyacinthus orientalis plant, they identified 8 nucleus as like same as like the ovule, female gamete. Same as like the female gamete. Usually you all. Tomorrow we will discuss, next class we will discuss the ovule structure, megasporangium structures. How the megasporangium, how it develop, how it get mature. Okay. Thank you, sir. Okay, sir. Right. Okay, sir. It's over. Very, very important. Examination point of view, very important. Anther structure, microsporogenesis. Okay. And types of the anthers that are very, very important. Okay. Pollen tetra types. Okay. Tetrahedral, decussate, bilateral, linear, T-shape. That also very important. And the special type of the structures also very, very important. Okay, cyperus pollen and Nemak phenomena. Okay, bye.